Hey everybody, in this video we are going to talk about genetically modified organisms. And I don't know why I always say we are going to talk about something, because these are videos and I'm not getting any feedback during the video. So, I am going to describe genetically modified organisms to you. Well, also known as GMOs. And as we all know, humans are, pretty good at making genetically modified organisms, but humans aren't the only ones to make GMOs. Other organisms can do this, and one of the most well-known or well-studied organisms other than humans that can make a genetically modified organism is called Agrobacterium tumefaciens. And I'll most likely just call it Agrobacterium. Now, this is a gram-negative bacterium. For those of you who have recently taken microbiology, that might be important to you or meaningful. So it is a plant pathogen. So many of you might not realize this, that there are lots of pathogens of plants. Bacteria and fungi are some of the worst pathogens. Viruses also. And Agrobacterium, as you can probably guess, it's a bacterium. I already said gram-negative bacterium is a plant pathogen of plants of 93 different families of plants. So some plant pathogens they're obligate biotrophs, we call those, and they have to be associated with their host. They can't live apart from their host. Agrobacterium is not one of those types of pathogens. Agrobacterium is happy in soil, happy in a free-living state in the soil. So it can be free-living or it can be associated with its host plant. So something interesting about Agrobacterium, because we want to know how Agrobacterium gets to the plant. Well, it has these little flagella that are arranged about 1/3 of the way. If you have two polar ends, well, 1/3 of the way from one end, you'll have what are called circumthecal flagella. And these things will help Agrobacterium get to a plant host. So also, now let's say I'm not very good at diagramming plants. Oh, I'm not very good at diagramming anything yet, but I'm working on it. Now, I just gave you guys a bonus activity in spring 2020 to watch some population genetics videos from Sal Khan of the Khan Academy. Now, he is the master at this sort of thing, and maybe someday I'll be able to do videos or make videos as good as his. But for now, we'll make do with what I'm doing this semester, hopefully. So, okay, so here's a plant, and we have some roots here, and, maybe an insect comes by and munches on, one of these roots or a root hair or something, and when that happens, it leaves a wound behind, and that wound oozes certain chemicals. Certain chemicals leak from the wound site. And Agrobacterium is really good about sensing those chemicals that leak from a plant wound. And it can move through a process called chemotaxis, towards the wound site. And you can imagine chemotaxis works by it's following the chemicals. And as the chemical concentration increases, which it's going to do near the wound site, the closer to the wound site, the higher the concentration of those chemicals, well, Agrobacterium can make its way to the wound. And when at the wound, it can crawl inside the wound and attach to a plant cell. So I'm going to diagram Agrobacterium like a rod-shaped E. coli cell here. So I'm not sure if it's oval like I did in one diagram, which if you see the notes, you know, one of our diagrams that I found on the internet had it, and it is an oval shape. And some other ones had it more like a rod-like shape like this. What's this, a rod-like shape? Yeah, I would call this is a rectangle. I should say rectangle-like shape. So this is going to be Agrobacterium over here. And this is going to be the plant cell. And let's say this is the plant cell wall and membrane. We'll say the wall and the membrane right here. And Agrobacterium is up against the wall here, and I could have diagrammed it closer, but I didn't. So the plant cell is much bigger, and I'm not going to diagram the whole thing. I'm just going to show this wall and membrane right here. So Agrobacterium will form what is called the T transporter complex. And this will be like a tube that extends from the Agrobacterium through the plant cell wall and the plant cell membrane into the plant cytoplasm. And here, you know, I'm going to diagram the Agrobacterium chromosome, which is very long. We could look it up if we wanted to. So let's just assume the chromosome is up there, but we are mostly interested in the TI plasmid, which is a pretty big plasmid, but nowhere as long as the chromosome. So these are not to scale, obviously. So this plasmid is called the TI plasmid, for tumor-inducing plasmid. And we're going to see that Agrobacterium, when it infects plants, it can cause tumors in plants, which are also known as galls. So these little points right here on this plasmid, we're going to call those the left border and the right border. Now, during the infection, what Agrobacterium does is it transfers a single strand of DNA from the TI plasmid between the left border and the right border. This is going to be called the tDNA. So one strand, not both strands, one strand is transferred through the T transporter complex into the plant cell, and then it's imported into the nucleus. Let's say this is the nucleus, and it is integrated into the plant genome. So why would Agrobacterium, you know, make its way through a plant wound, attach to a plant cell, take a piece of DNA, put it into the cytoplasm, help it go into the nucleus? There are going to be some proteins that are mediating this process, and then put it into the plant genome. What purpose does that serve Agrobacterium? Okay, well, we're going to see that in the next, we'll call this slide here. Well, to see that, let's take a closer look at the TI plasmid, the tumor-inducing plasmid. Oh, that's a pretty good circle. I'm getting pretty good at this. So we have the left border here and the right border there. And remember, a single strand of DNA is transferred over into the plant cytoplasm and then to the nucleus, and it's integrated into the plant genome. And at some point, it's going to be made double-stranded. It's going to be stably integrated into the plant genome. So every time that cell divides, the daughter cells will have a copy of that tDNA. So between the tDNA, there are three types of genes. So we have a gene for auxin, which is a plant hormone, a gene for cytokinin, which is another plant hormone, and then we have a set of genes, I believe it's a set of genes, involved in opine anabolism, which is a fancy word for synthesis or biosynthesis. Now, opines are small nitrogen-containing compounds that only some organisms can use. Very few organisms actually can metabolize opines. Agrobacterium happens to be one of the organisms that can use opines. The plant cannot use opines. So we'll talk more about that in a moment. Okay, so those are all the important genes between the left border and the right border. Those are all the important genes in the tDNA. T is for transfer DNA. Now over here on, let's say, this side of the TI plasmid, we're going to have a set of genes in this region here. This isn't one long gene. This is a set of genes, so many genes in here, that are the virulence genes. Now, these are the genes that, are responsible for, making the proteins that take the single-stranded tDNA, transport it over to the cytoplasm, help it get into the nucleus, get into the genome. So that's what the virulence genes are needed for. So they're needed to transfer the tDNA, and they might even be part of the T transporter complex. So over here, we're going to have the origin of replication. Then over here, we're going to have a set of genes involved in opine catabolism. So that's breaking down opines. So previously I said very few organisms can metabolize opines or can catabolize and break them down and use them as a nutrient source. Well, Agrobacterium is one of them because it has genes needed to catabolize those compounds built right into the TI plasmid. Okay, so what does all this do? Why is Agrobacterium putting a significant amount of effort into transferring this to the plant cell? Well, so what it does, auxin and cytokinin, what they do is they cause plants to start dividing. You have a plant that's not dividing, it will cause that plant to start, that cell, I'm sorry, did I say plant earlier? So these will cause cells to start dividing. So let's say here, this is a plant cell. This is agro. Okay, so this plant cell now has the tDNA in its genome. It starts making auxin and cytokinin. So these are all the other plant cells around there. And as a result of all this auxin and cytokinin being made, all these plant cells start dividing. They all start dividing. And then this one's going to divide, right here that has the tDNA. And let's say everything that, every daughter cell that comes from this one is also going to have the tDNA, and they're also going to make auxin and cytokinin. And so what's going to happen, let's say this is a little plant right here. And let's say the infection happens somewhere down here. What's going to happen, we're going to get a big tumor right here, also called a gall, which is just some really abnormal growth coming off of, say, the stem of the plant. And so this abnormal growth is caused by the auxin and cytokinin. So all the plant cells that weren't supposed to be dividing are all of a sudden they start dividing. And why does Agrobacterium like that? Well, because these cells right here, well, one has got a bigger shelter now for more Agrobacterium to be protected from the environment. But every cell that has the tDNA is also going to be making opines. And that's a nutrient that only Agrobacterium can use. So what we end up with is a giant gall that makes lots of opines that only Agrobacterium can use. So essentially Agrobacterium has made itself a perfect little home, or niche would be the biological word. I like home though, right? Perfect home for itself to make lots of Agrobacterium, lots of daughter cells. It's got a food source that only it can use. So, you know, it's really happy. And it did this by genetically engineering the plant genome. So Agrobacterium tumefaciens is an organism that makes GMOs just like humans. Humans make GMOs to, you know, make things better for us, ideally. Agrobacterium does the same thing. So humans also like one thing I should point out. So if you look in the notes, there's some details on the virulence genes, the different names of the proteins and the roles that they're thought to have in transferring the tDNA. We don't really need to worry so much about that. What we're interested in is, you know, why does the gall form? What the auxin and cytokinin genes do, what the opine gene does, what the opine anabolism genes do, what the opine catabolism genes do, and of course origin of replication. We know what that's for, right? The plasmid wouldn't be replicated in Agrobacterium if it didn't have an origin of replication. So humans like to make GMO plants, or I should probably say GM plants, genetically modified plants, but it's not so easy, to do that. So scientists have come up with a way to make it easier by using Agrobacterium. Okay, so how do we use Agrobacterium to genetically modify a plant for use by humans? Well, it essentially, you know, we modify the TI plasmid. So we know a lot about working with plasmids from, you know, decades of research on E. coli. And a lot of the techniques are similar for working with plasmids from Agrobacterium. So if this is the tDNA region with auxin and cytokinin and the opine genes, and over here are the virulence genes, and over here, what did we say, were the opine catabolism, right? Yeah, opine catabolism genes, anabolism genes up there, the origin right here. So what scientists have done essentially is, you know, take these guys out. We don't need these anymore. We can grow Agrobacterium tumefaciens in the lab. In the lab, it doesn't need its TI plasmid, so we can take its TI plasmid out, and we can modify it, and then we can put it back in. So, and when we modify it, one of the first things we can do is keep the left border and the right border, because those are needed to transfer tDNA, and replace everything between those with a multiple cloning site. We saw multiple cloning sites, I think, in lecture 23, maybe it was 22. Multiple cloning sites, what are those? So those are sites that have lots of different restriction enzymes. And those restriction enzyme recognition sites. And this will allow us to put lots of different genes in between the left border and the right border. any gene we want to put in the plant, we can just put now in the multiple cloning site between the left border and the right border. So we can remove other things from this plasmid too to make it a little smaller, to make it easier to work with. Small plasmids are easy to work with because they're easier to get back into the bacterium after you take them out. The bigger the plasmid, the harder it is to get back into a bacterium using, electroporation or the calcium chloride competency procedure that we talked about in a previous video. But we need to keep the virulence genes in there. And for those of you who are advanced in genetics already, maybe you're working in a genetics lab, there are also ways to split these genes into two plasmids, like a helper plasmid and a TI plasmid or something like that. But we're just going to keep everything on one plasmid in this description of the conceptual or the concepts behind agrobacterium mediated transformation for research purposes. Okay, so we need the virulence genes because the virulence genes are the things that make the proteins that are needed to transfer the tDNA. But you can probably bet we don't need the opine catabolism genes anymore. If we're going to make a genetically engineered plants for our uses, well, the TI plasmid doesn't need opine catabolism genes. So we can replace those with anything. But keep in mind, anything we put on this side of the plasmid is not going to be integrated into the plant. So we can put, let's say, an antibiotic resistance gene here. Why would we want an antibiotic resistance gene in the TI plasmid? Well, once we take the TI plasmid out of Agrobacterium and we try to put it back in, well, we're going to need a way to know if it's inside the bacterium. So the antibiotic resistance gene will let us know that, oh, okay, we succeeded in, we took it out, we succeeded in getting this TI plasmid back into the bacterium because it's now resistant to, we could say, ampicillin. And there's some others, kanamycin and geneticin, I think. So that might be one reason why you would replace these opine catabolism genes with an antibiotic resistance gene. And here's the origin of replication. We would need one of those. So let's take a look at a specific example that is commonly used in research. Am I still recording? Yep. So there is what turns out a really important gene in plants, important for a number of reasons. We're going to call this EPSP synthase. And there are most plants have the same allele, and the allele is going to be, we'll call it allele S. And there's also allele R. But most plants have allele S. So, and what is EPSP synthase? So it stands for, well, this long, complicated name. And I do love chemistry. Someday I'd like to go back and take organic chemistry. I know a lot of my students and a lot of you are taking organic chemistry right now, and really fascinating. And I didn't appreciate it enough when I was an undergraduate student. So 5-enol pyruvyl shikimate. 3-phosphate. So that's the EPSP part, 5-enol pyruvyl shikimate 3-phosphate. And then we have the synthase. So essentially this compound right here is used by plants. Used by plants to make what? 3 amino acids. So 3 amino acids are produced only if the plant can make this compound right here. So tyrosine, these are all aromatic amino acids. Alanine and tyrosine, phenylalanine, and tryptophan. Okay, so only plants use EPSP synthase to make this to make these guys right here, these amino acids. Humans, animals don't use this. Which is why there's a herbicide called glyphosate, which is the herbicide in a very famous, well, some people will call it notorious, but I think farmers love it. Farmers love this herbicide. Glyphosate, it's found in Roundup. And what glyphosate does is it shuts down the sensitive allele of EPSP synthase. So the plants can no longer make these amino acids. So the plants die. So that's why glyphosate is a herbicide. So if you wanted to make a plant that was resistant to the herbicide, what you would do is you would need to give it allele R. I don't know if I should call these alleles. I guess different versions of the same genes, because I think this R version of the gene isn't necessarily naturally found in plant populations, but it might come from an microorganism that is resistant to glyphosate. I don't really know the history of where the resistant version of EPSP synthase comes from. But in the notes, we distinguish between the sensitive version, EPSPS, and EPSPR. So we'll call them alleles. Yeah, we'll stick to that. Different versions of the same gene. So yeah, we can call them alleles and be safe. So, okay, so how can we make a glyphosate resistant plant? Well, kind of already went over that, right? You can probably guess if you were paying attention. We're going to take our recombinant TI plasmid. Recombinant means DNA from two different sources. And why is this recombinant? Well, because we already took out all those important genes that the agro needs for infection in nature. Well, it doesn't need it in the laboratory. And we replace those with a multiple cloning site. And we have antibiotic resistance here. And we have virulence genes over here. And what we're going to put in here is gene for EPSP synthase. We're going to put the resistant version in there. Okay, so we're going to put this gene in here, and we can use restriction enzyme based insertion into the multiple cloning site, which we talked about in a previous video. And then we need to get this into Agrobacterium, an Agrobacterium that has been cured or relieved of its natural TI plasmid, and we can put this back into Agrobacterium. And then we need to make a large culture of the Agrobacterium with the TI plasmid. And how can we know that the Agrobacterium has the TI plasmid? Well, we can add antibiotic, whatever antibiotic resistance gene is here, we can add that antibiotic to the culture. So the only way agro can grow is if it has this recombinant plasmid. And then, you know, there are a number of ways to get the agro, I guess, in sort of mix it with plant cells so it can genetically engineer those plant cells for us. And what I mean by genetically engineer is transfer the tDNA containing the EPSP synthase gene into the plant genome. One of the easiest things to do, this works with many plants. So let's say this is our Agrobacterium culture inside a beaker. And then here's our plant. And let's say here are the reproductive parts of the flower, reproductive tissues. Well, what we can do is dip this flower into the agro culture. And the agrobacterium will mix with the reproductive tissues, and then when that plant produces seeds, some of those seeds are going to have the tDNA integrated into the plant genome. So when we plant the seeds, here, let's say this is a little, some soil in a tray in a greenhouse. So they will grow up, make new plants. Now, not all of them are going to have the tDNA in the genome. So how can we know? How can we know which plants, let's say we get thousands of seeds or tens of thousands of seeds. How can we know which one or which of the plants have the tDNA inserted into the genome? Well, we could take our bottle of glyphosate herbicide, and we can spray the plants, right? So we're spraying the plants. This is me moving the bottle. And the plants that survive are the plants that have the tDNA integrated into the genome. So we have our GMO, genetically modified organism or genetically modified plant that is resistant to glyphosate. So glyphosate. A lot of people are against this herbicide because it's so widely used in conventional agriculture. I guess farmers, I have some pictures in the notes that show you a field of, I think it's soybean maybe, or corn, I forget, but it shows you like the before and after with glyphosate. And this is a field that's growing crops, and you can see before treatment with glyphosate, there are weeds everywhere. After treatment, there are no more weeds. So you can imagine this is a really powerful way to remove weeds from a field without injuring the crop plant. And I guess it increases yields and all things like that. And it's supposed to break down in the environment relatively quickly. And it is, like we said, it's supposed to be non-toxic to animals. So one of the things I'm interested in is, it's non-toxic to us, theoretically non-toxic to us. All the research says, okay, it's not cancerous, at least in the doses that we would be exposed to it. And it's not toxic. But, what about our gut microflora? So we have billions of bacteria, right? Or maybe up to a billion, I don't know. But very, very many microorganisms living in our digestive system. And how does glyphosate affect those? So I'm interested in learning more about that. I think there are researchers investigating that. How much glyphosate is if you eat genetically modified crops or stuff, foods that are from genetically modified organisms that have been modified to be resistant to this, how much glyphosate is found, how much glyphosate residue is found in those crops when we consume them. I don't know those numbers, but it'd be really interesting to know if there are detectable levels there, and if there are, what do those levels do to gut microflora? So there's lots of stuff to research with respect to this. Okay, so what we want to do, so there's a question in the notes. What would you do? Okay, so what if you want to make a different genetic modification to plants? What if you want to put a gene into a plant that doesn't have to do with herbicide resistance? Well, one thing you could do is you could do both, right? So you could use herbicide resistance here. So let's say EPSPR there, and then you could use whatever other gene you wanted to right next to it. So they would both go into the genome. And let's say a gene to make vitamin A. I don't think there's probably one gene that you can do that, but I think there's a rice called golden rice. I guess rice has very low levels of vitamin A, and scientists have succeeded in making a version of rice that has high levels of vitamin A. I've actually never seen it, but I think it's supposed to be a golden color. But this is one thing you could do. You could just attach the gene of interest, downstream of the herbicide resistance gene, and then, okay, you can select for plants having the tDNA based on resistance to the herbicide, and they should also have the copy of vitamin A. And then there are other things you can do. You can use antibiotic resistance genes. I think hygromycin resistance. And then when you do that, you need to find ways to keep this in the genome while removing this. And there are lots of things, highly technical things you can do to, get one gene in and remove the other. But those are beyond the scope of this course. So